The Príncipe Felipe Research Centre focuses on two important lines of research linked to proteins that are used directly to cure diseases, through the creation of biopharmaceuticals from peptide synthesis, as well as via treatments that reverse the harmful effect of various genetic modifications. Dr. Orzáez leads these two lines of research at the Peptide and Protein Chemistry Laboratory, where expertise and the most advanced equipment merge.
So we have the possibility to synthesize longer peptides. This is a clear advantage for our users because they can work with longer parts of the protein, and this gives the opportunity to achieve more complex projects.
This technology enables the synthesis of longer peptides and those with more complex sequences, and offers comparative advantages over the traditional chemical system, which came up against problems during its synthesis process.
The microwave techniques avoid the aggregation of the peptide during the synthesis and give us the opportunity to synthesize longer peptides with better efficiency and higher quality in the final product.
In this case, this team's expertise is combined with high technological capacity to secure better final results at all levels.
So we have the possibility to do microwave synthesis of peptides. This gives the possibility to perform longer peptides and also with high purity and quality.
Another comparative advantage of this research group is their specialization in hydrophobic peptides, which, given their complexity, cannot be studied in other centers. Now, this group is seeking new challenges in a field that is expected to be a crucial source of biopharmaceuticals in years to come. It is possible for them to mimic the function of a protein or antagonize it by using a fragment of the same protein. This creates the prospect of using them as pharmaceuticals with benefits in the mechanism of action.
Next step is trying to adapt the Orzáez in order to perform high-throughput screenings, in order to improve the time that you need to characterize a mutation and to look for new treatments in less time.
The second line of research entails discovering the effects of genetic mutations on proteins and even modifying them in order to restore their function.
In our lab, we have the capabilities to introduce these mutations in the gene and to study which are the consequences on the function of the protein.
This field of research opens up countless possibilities, considering that many mutations linked to diseases are being identified in today's genomics era. That said, it is necessary to identify the precise effects of these mutations and determine how they can be reversed.
We have the capability to understand if the mutations are affecting functions of the cell, such as cell cycle, cell survival, cell migration, or if they are affecting the localization of the protein inside the cell. And we can study which are the consequences related with the disease of these mutations. And then we can design how to fight against these consequences to obtain new treatments, for example, for diseases.
This procedure is being used in a lot of tests nowadays, but its purpose is to increase the number of phenotypes that might be linked to the disease. Despite this, they are also setting themselves a challenge that would see them making progress at a much quicker rate.
We have now the capability to perform the assays by only one by one, to study how one mutation produces changes in one protein, and what we want to do in the next step is trying to adapt these assays in order to do high throughput screenings and to understand how many mutations induce changes in the function of many proteins at the same time. This would give us the possibility to improve the time that we need to detect which is the cause of disease and to design new treatments for the disease.
In this field of genetic mutations as well as in peptide synthesis, the results of their research are progressing in line with the technological developments that are underway. By incorporating them into their procedures, results are not only improved but can also be obtained in a lot less time.